Okay, so how do we look at this? Well, there's some neat little experimental tricks. I'm going to describe one kind of widely used technique, set of techniques that allow us to detect changes in chromatin structure as time goes on, or prior to transcription, or during transcription, or what have you. And we call these experiments nuclease protection experiments. And these are important tools for getting an idea of changes in chromatin structure, such as prior to transcription, things like that. Now, how does this work? It works like this. Nucleases, we're talking about enzymes that digest nucleic acids. Specifically, we're looking at DNA digesting enzymes. Now, there's a whole bunch of these DNases around. Some of them are very aggressive and will practically attack just about any DNA anywhere they can get their hands on. Others are not quite as aggressive, and their action can be blocked if the DNA is covered by proteins. So what we do is you take chromatin, okay? And nucleases, DNases. Areas of chromatin that have few or no proteins on them are going to get preferentially attacked by the nuclease. So they get chopped up. Areas that are covered by proteins are going to be more resistant. They may get cut eventually, but they're going to be highly resistant to nuclease attack. So now what we do, we're going to chop the chromatin up into pieces. The pieces are going to be, the places where the cuts occur are going to be the areas that have either no protein or have proteins that are fairly weakly attached to them. So now what we do, we take this and we separate the DNA and the protein. And then we run the DNA on gels to separate by size, run the proteins on gels. So we can see, for instance, on electrophoresis gel, we might see fragments of different sizes representing the pieces that are intact and after we've cut things up. We look at the proteins and we'll see certain kinds of proteins. Okay, now what we can also do is we can take these fragments of DNA. These are the fragments that have not been attacked by the nucleases. These are areas that are protected from nuclease attack by the proteins that were covering them. And this gel shows the proteins that were covering these sequences.
So they had to be on a different side, right? They were not kind of in the vulnerable area.
Yeah, exactly. Because the vulnerable area has gotten chewed up and destroyed. It's a bunch of nucleotides at this point. Okay, so, what would be a good analogy? Okay. Well, let's suppose we had a wire, okay? And we coated some of the wire with a thick layer of like Teflon or something like that. And we had other layers of the wire exposed. We had different pieces of, different lengths of Teflon covering the wire. Long piece here, short piece here. Now we take that wire and throw in some really nasty acid. The acid is going to attack and chew up the wire that's not covered by the Teflon. So now you take this thing out. So you have a bunch of Teflon pieces with wire in them. The bare wire is completely dissolved in the acid. You wear gloves when you do that, by the way. Okay, and now what you do is you tear the Teflon off the wire, run the Teflon in gels to see what it looks like, and then you measure the length of the pieces of the wire that was protected by the Teflon. Is that a fairly good analogy there?
Yes.
Okay, all right. So these are the pieces that are protected from nuclease attack. These are the proteins that are doing the protection. Now, if we want to go more, we can sequence these. Each piece, we can figure out what the DNA sequence is. We can do various kinds of analysis on these proteins. So the key thing about this, these nuclease protection experiments tell you what sequences of chromatin are covered by proteins and what proteins are covering them. Needless to say, you're going to find a lot of it are these 146 base pair of fragments, and the proteins associated with them are the histones. But you also find other things too. Okay, so that's the basic idea behind a protection experiment. Now, what kind of cool things can you do with it? Well, let's do this. Let's take some cells that we can induce transcription of certain genes with at will. For instance, steroid hormones often lead to transcription of numerous different genes. That's how they work. Steroids work by regulating gene expression, usually causing transcription of a whole bunch of genes. Okay, so we can take some cells. All we have to do is treat them with steroids. Within about 20 minutes, you start seeing new proteins. You start seeing genes transcribed. The question we have here is, what's going to happen to the chromatin before treatment and during treatment, and finally when we actually start seeing transcription occur? So let's do nuclease protection experiments. We stimulate the cells with hormones. We take a control. We stimulate the cells with hormones. We wait 5 minutes. We wait 10 minutes. Each time we do that, we isolate chromatin, chew it up with nucleases, run the proteins, and run the DNA fragments on gels. And we see what's going to happen. Well, what happens here let's just using two examples. One point here. Beforehand, we see, things like a ladder of 146 bases. We see a number of highly protected regions. And for the most part, most of these protected regions are the regions covered up or wrapped around the histones to make the nucleus cells. But then, shortly, maybe about 10 minutes after stimulation, but prior to transcription actually taking place, now what we start seeing is these things are getting broken up. We'll still have some, but we see a lot more fragments around than we used to.
What does that say on top of the
Here?
next to the other side?
Here.
Right side.
Yeah, that.
Yeah.
Oh, 10 minutes. The little slash means minutes. Okay, what we see here now are new cuts.
It's okay. It's snowing outside.
Some of these new fragments are going to happen extremely quickly. Just touch them with nucleases and you see these fragments. So we have some of these things that are sometimes called we often call them hypersensitive sites. These are places that once were protected and now their throats are laid bare to the wolves of the nucleases. So they get cut quick and fast and easy. Now we can run the proteins and we may see differences in protein complement too. Maybe we see additional proteins that weren't there before. Maybe we see some proteins that were there before that aren't present anymore. These types of experiments give you an insight into proteins coming into the chromatin, proteins leaving the chromatin, areas of DNA that are not tightly bound to protein anymore and therefore getting attacked by nucleases, areas that are still protected, and so on. But a consistent thing is just before transcription, you see a whole bunch of these formerly protected areas now getting attacked viciously by the nucleases. That's our hypersensitive sites. And that's telling you that something is happening. Either proteins are moving off of certain areas, or they're being replaced, or they're simply not binding as tightly. So it's telling you there's lots of changes in the chromatin occurring prior to transcription. And more parts of the DNA are either exposed or only weakly protected, and now they get attacked when they weren't getting attacked before. So that's saying, once again, experimental evidence, chromatin structure is being altered in some way where it's not binding DNA as tightly prior to transcription. Now these kinds of experiments are giving us the first insights. What's being protected, what's no longer being protected, what proteins are involved in. Now, of course, we've taken things a lot further. We find one of the most important things that happen are what's going on to the histones before transcription takes place. So in just a moment, we're going to take a look at that.